Hi everyone, so this is going to be a EOC Genetics, which is heredity review for the end of course exam for biology. Let's go ahead and get started with some vocabulary recap. So some vocabulary recap, we need to remember what a genotype is. A genotype is a letter combination. So basically it's represented by two alleles. That means we're going to represent something we cannot see. So genotype, think genes. Phenotype, think something we can see, physical feature or physical appearance. And that's because of the genotype. So based on the genotypes you have in your DNA that you cannot see, you are going to express the phenotype. So that is what you can see. Then we have traits. Traits are going to be characteristics that are passed from parents to offspring. A good example could be eye color. Alleles are the possible forms of the trait represented by letters, again, lowercase and of course uppercase. An example could be uppercase and lowercase. That gives us our blue eyes, which would be our phenotype. So the alleles are usually going to be our genotypes. Dominant versus recessive. So a dominant trait is going to be the stronger of the two alleles, which is represented by the capital letter. Recessive will be the weaker of the two traits or the two alleles, which means it's usually represented by a lowercase letter. Homozygous, heterozygous, and then of course males versus females. Homozygous, usually what you're going to see with homozygous is we have two of the same alleles or letters, and they're either going to be capital for homozygous dominant, or they're going to be lowercase for homozygous recessive, meaning it's the same trait. Heterozygous. Heterozygous is going to be different, which means it is a different combination. So we could see an uppercase letter, which would be the dominant trait, and a lowercase letter, which would be the recessive allele. Males versus females. Males contain an X from mom and a Y from dad. Females are going to contain one X from mom and one X from dad. So males are going to be XY, while females would be represented as XX. Let's go ahead and look at Punnett squares. So Punnett square is just a visual representation of a test cross, and it's going to predict the possibility of genotypes and phenotypes among offspring. So if you look across the top, we have two types of crosses. We have a monohybrid cross, which is going to look at one trait. And then we have a dihybrid cross, which actually predicts two traits. So if we look at our Punnett square that we're given, if we have a monohybrid cross of pea plants that are heterozygous, which is capital Y, lowercase y for pea color, what would be our genotypic ratio? So if we were to look at a Punnett square, and we're going to go back, let's erase this because these are offspring that are heterozygous. We want to look at parents that are heterozygous. So we're going to see we have a capital Y with a lowercase y and a capital Y with a lowercase y because heterozygous means different alleles, so capital and lowercase. So in the first box, we'd have capital Y, capital Y. Second box over, we'd have capital Y, lowercase y, capital Y, lowercase y, and lowercase y, lowercase y. So on our Punnett square, we're going to see 3 out of the 4, or 75%, are going to be dominant, which is going to be yellow in this case, while only 25% of the peas will be green, which will be 1 out of the 4. Dihybrid crosses, that's when we see two traits being compared. So for pea plants that are heterozygous for pea color, that's going to be capital Y, lowercase y. And pea shape, which is capital R, lowercase r, the first thing you're going to do is FOIL. So first, outer, inner, last. When we do our genotypes for the parents, we're going to see capital R, lowercase r, capital Y, lowercase y, and that's going to be the same for both of our parents. The phenotypes would be round and yellow. So here's our Punnett square if we were to do a heterozygous cross between two parents, and you're going to see the phenotypic ratios are 9 to 3 to 3 to 1. So there are 9 round and yellow, 3 round and green, 3 yellow and wrinkled, and 1 green and wrinkled. Mendel's laws. So Mendel's laws follow the law of segregation that states the alleles are separated from each other during gamete formation, which is meiosis, and recombined at fertilization. So meiosis results in the formation of gametes that contain one allele of each trait, which means it's haploid. Then fertilization combines the gametes, which of course that's the egg and sperm, resulting in a diploid cell containing 2 alleles for each trait. One trait comes from each parent. Another one of Mendel's laws is independent assortment. The law of independent assortment states that allele pairs separate independently during gamete formation. Thus, traits are passed down to offspring independently of one another. Increasing genetic diversity, of course, among organisms that occur during sexually reproducing organisms. So we're going to see that independent assortment and segregation actually allow for, in meiosis, prophase 1 of meiosis 1, that's going to allow for more genetic diversity because it's shuffling all of the genetic information. So segregation, we see a figure on the left where all of the chromosomes are separated, aka the traits. And then independent assortment, we can see that on the right, how the traits independently assort from one another. Let's go ahead and check out a little example for the patterns of inheritance for the law of dominance. So the law of dominance states that any organism with at least one dominant will express the dominant form of the trait. For example, cross 2 heterozygous pea plants for height. We have tall, and then we have the short trait. When you fill in all of your Punnett squares, you're going to see, similar to what we were looking at with pea plant color in our first Punnett square, that the genotypic ratio, remember that's the genes, we're going to see there are 1 out of 4, capital T, 2 out of 4, or 1/2 for capital T, lowercase t, and 1 out of 4, lowercase t, lowercase t. What percentage of the offspring are tall? We have 1, 2, 3, so 75%. Each of these boxes would be 25%, and we know that 25 plus 25 plus 25 plus 25 gives us 100, and it would equal a total of 100% of the offspring. According to this principle, which genotype can never produce offspring that express a recessive phenotype? That's going to be our homozygous dominant. They don't have the recessive trait, so they cannot produce recessive offspring. Some alleles that are neither dominant nor recessive, it's called codominance. And this occurs when the phenotypes produce both alleles and they're clearly expressed. For example, human blood type is a form of multiple alleles. And codominance, we have 4 different blood types. We have type A, we have type B, we have type AB, and we have type O. So type AB, that's going to be our codominant trait because this person or people that have type A, B will carry both the type A blood and the type B blood. So type A and B are going to be dominant together, and type O is going to be the recessive phenotype. If a boy has a blood type O and his sister has blood type AB, what are the genotypes and phenotypes of their parents? So we have a type O child. We have a sister who has type AB. That means one parent has to be A, the other parent has to be B. Because how can we see type AB blood? Now, their son has type O, which means in order to see the recessive trait, we are going to see that the parents are carriers of type O. Otherwise, we would not see that recessive trait. So the phenotypes of the parents would be type A, carrier O, and type B, carrier O. So we don't know which parent is type A. We don't know which parent is type B, but for type A, we would see there's the genotype for A, carrier O, and here's the genotype for B, carrier O. Sex-linked traits, they're going to be located on the X chromosome, okay? So yes, they could be on Y, but not typically. They're typically going to be X-linked traits. So they're carried on the X chromosome. So females rarely actually express a trait that is on the X chromosome because males have two X chromosomes. So usually we're going to see females rarely express an X or sex-linked trait, only if it's homozygous recessive for the female. So if we were to look at a female that has hemophilia, that means she would be recessive for hemophilia. And let's say the father has hemophilia, and this could be for color blindness too. We're going to see here's two daughters that have hemophilia. But if one of the parents, let's say the mother, is just a carrier of hemophilia, so the lowercase h, we're going to see now only one daughter has hemophilia. So what are the genotypes that we can see for a female? We can see X capital H, X capital H, X capital H, X lowercase h, and X lowercase h, X lowercase h. So this would mean they have the trait or they have this deficiency, whatever is on the X chromosome. Where males, because they only have one X chromosome, they are going to either have the trait or not have the trait. Okay. Let's go ahead and cross a red-eyed male and a white-eyed female. Now we're talking about Drosophila fruit flies. So we have a red-eyed male and a white-eyed female. Red is going to be capital R, white will be lowercase r. We're going to see we have a red-eyed female. We have a white-eyed male. We have another red-eyed female and another white-eyed male. So all of the possibilities here for our offspring would be for females, they're going to be only red eyes. For males, they're only going to have white eyes. Now, multiple alleles. Multiple alleles are when genes with more than two alleles. If you look, we have a brown, a black and white, and then a white fur hair. So now we're going to look at an individual that only has two alleles, but there are many more than two alleles possible for the population, which is a gene pool. And it's represented by I for humans. Again, refer back to blood type for this example. Polygenic are when there are many different types of traits that are controlled by two or more genes. So basically, when we think about a polygenic, you can look at eye color. So how often a variety of phenotypes that we can